Good morning, all of you. I'm going to repeat again how the 2,3-bisphosphoglycerate stabilize the T form of the hemoglobin. Okay, T form of the hemoglobin. 2,3-bisphosphoglycerate stabilize the T structure of hemoglobin. Okay, T structure of hemoglobin. A low partial pressure of oxygen in the peripheral tissue. Whenever the peripheral tissue partial pressure is very low, okay, this situation promotes the synthesis of 2,3-bisphosphoglycerate in erythrocyte. Okay, in the glycolytic, from the glycolytic intermediate 1,3-bisphosphoglycerate. Okay, already it was taught in carbohydrate metabolism class, Rappaport liberating cycle. Okay, Rappaport liberating cycle. Okay, how the 2,1,3-bisphosphoglycerate, it is bypassed to the formation of, for the formation of 2,3-bisphosphoglycerate. So there is a formation of the 2,3-bisphosphoglycerate always whenever the partial pressure of the oxygen, it is very low in the peripheral tissue. Okay, usually in the high altitude. The hemoglobin tetramer binds one molecule of BPG, bisphosphoglycerate, 2,3-bisphosphoglycerate. Okay, not 1,3, 2,3-bisphosphoglycerate. Hemoglobin tetramer binds one molecule of the 2,3-bisphosphoglycerate, not 4 molecule, only one molecule. Okay, not like him, isn't it? Whole the hemoglobin tetramer means 4 polypeptide chain all together binds only one molecule of the 2,3-bisphosphoglycerate in the central cavity. Okay, central cavity formed by its 4 subunit. Okay, formed by its 4 subunit. 4 subunit means 4 polypeptide chain. Okay, 4 polypeptide chain, they form the central cavity. Okay, where 2,3-bisphosphoglycerate is attached when the partial pressure of the oxygen is very low, very much low in the peripheral tissue. Okay, the space between the H helices, the space between the H helices of the beta chain, H helices of the beta chain means beta polypeptide chain, okay, 2 beta 2 alpha. So that is the H helix, 75% of the residue in the under helical structure. Okay, helical structure. Almost the all hemoglobin molecule, there is a formation of the right-handed helix, right-handed helix in the 75% structure. Okay, 8 right-handed helix. Okay, the space between the 8 helices of the beta chain lining the cavity is sufficiently wide to accommodate, okay, 2,3-bisphosphoglycerate only when the hemoglobin is in T state. BPG form the Sol bridge with the terminal amino group. Okay, 2,3-bisphosphoglycerate form ionic bond, that is the Sol bridge with the terminal amino group of both beta chain. Okay, of the both beta chain, terminal amino group of the both beta chain via valine non-helical part. Okay, non-helical part of the A helix. Okay, in the A helix, A also the right-handed helix. Okay, there the presence of the 8 number of helix. Okay, that A number A helix, there is a non-helical part exist. Okay, so in the via valine non-helical A1 and with lysine EF6. Okay, lysine EF6. Lysine EF6 means amino acid lysine present in the E and F helix in the 6th position. Okay, and histidine H21. Okay, in the H helix 21 amino acid histidine. Okay, along this amino acid the BPG form the salt bridge with the amino terminal group. Okay, of this both beta chain. Okay, the both beta chain amino terminal group, that the valine lysine EF6 and histidine H21. Okay, those amino acids are present in the amino terminal end of the both beta chain where BPG form the ionic bond or salt bridge. BPG therefore stabilize deoxygenated T state hemoglobin by forming additional salt bridge that must be broken prior to conversion to the R state. Residue H21 of the gamma, residue H21 is the histidine, usually in the beta polypeptide chain, but residue H21 of the gamma polypeptide chain present in the fetal hemoglobin, that is the alpha 2 structure is alpha 2 gamma 2, already I have mentioned. Okay, so residue H21 in the gamma subunit of the fetal hemoglobin is serine. Okay, serine rather than histidine. Okay, since serine cannot form the salt bridge in 2,3 BPG, so it is bind more weakly to fetal hemoglobin, okay, than to the adult hemoglobin. The lower stabilization afforded to the T state by BPG, okay, accounts for hemoglobin F having a higher affinity for oxygen than adult hemoglobin. Okay, as the histidine H21 is replaced by serine, okay, so serine cannot form salt bridge with BPG. Okay, so BPG bind weakly to hemoglobin F than as compared to adult hemoglobin. So it's lower the stabilization afforded to the T state, okay, T state by the BPG, okay, that is accounts for the hemoglobin F having a higher affinity for oxygen than hemoglobin A because of this replacement. This is the structure how BPG bind in the central cavity of the hemoglobin tetramer, okay, hemoglobin tetramer by forming the salt bridges to the amino terminal end of the both beta polypeptide chain via non-helical part valine A1, okay, non-helical A1 part of the valine, non-helical part of the A1 of valine, okay, lysine EF6 and histidine H21 of the both polypeptide chain. They form the central cavity where BPG attach by forming the salt bridge. Adaptation to high altitude. Physiologic changes that accompany prolonged exposure to the high altitude include an increase the number of erythrocytes and their concentration of hemoglobin and of BPG. Elevated BPG lowers the affinity of hemoglobin A for oxygen, which enhance the release of oxygen to the tissue. Okay, in the high altitude, okay, there is the increased synthesis of BPG. So there is a elevated concentration of the BPG, okay, which lowers the affinity of the oxygen towards the hemoglobin A, okay, which in each turn enhance the release of oxygen at the tissues. That means BPG doesn't allow the hemoglobin to go to the R form, isn't it? It allows the hemoglobin to remain in the T structure, T form. Okay, so that the oxygen affinity for the oxygen of the adult hemoglobin is very less. Okay, which enhance the release of oxygen at the tissue. Now, numerous mutant human hemoglobin have been identified. There are very much mutation of the hemoglobin. Okay, they very different types of, okay, various types of mutations occur in the adult hemoglobin. Okay, which results in hemoglobinopathy. Okay, hemoglobinopathy. Okay, so numerous mutant human hemoglobins have been identified. Okay, mutation in the gene, okay, this mutation may be lead to qualitative defect or quantitative defect, okay, either quantitative hemoglobinopathy or qualitative hemoglobinopathy or quantitative hemoglobinopathy due to mutation, okay, due to mutation. This mutation may be result in the either qualitative hemoglobinopathy or quantitative hemoglobinopathy. Please remember that, like that. Mutation in the gene that encode the alpha and beta subunit of hemoglobin potentially can affect its biological function. Okay, the gene that is responsible for encoding the alpha and beta subunit, alpha and beta polypeptide chain, that means, okay, polypeptide chain, that is a subunit also called as subunit of hemoglobin, potentially affect its biological function. Now methemoglobin, okay, In the earlier of the structure of the function as hemoglobin class, I have mentioned there is a difference between the oxyhemoglobin and oxygenated hemoglobin. Okay, deoxygenated and deoxyhemoglobin is different. Okay, there is a difference between the oxyhemoglobin and oxygenated hemoglobin, and deoxyhemoglobin and deoxygenated. Okay, these are the different terms. Okay, in the earlier of these classes I already mentioned. Now methemoglobin, also known as hemoglobin M. Hemoglobin M. I think about the methemoglobin you are taught in the HMP pathway, right? In the HMP pathway, you have, under the significance of the HMP shunt or pentose phosphate pathway, you are already taught about the methemoglobin. Okay, how methemoglobinemia occur, right? In the methemoglobinemia, okay, in the it will come as a short notes or it is very important. Okay, whenever you attend the clinics, this concept you have to remember. This concept is very important. Okay, the methemoglobinemia, the heme iron is ferric. Okay, heme iron is ferric rather than ferrous. Okay, heme iron is ferric rather than ferrous. Okay, methemoglobin thus can neither bind nor transport oxygen. Okay, when as soon as the iron become in the ferric state, that one valency becomes less. Okay, in the ferrous state, the iron has got 6 valencies. Okay, 4 valencies are occupied by the imidazole nitrogen of the histidine molecule. Okay, imidazole nitrogen of the histidine. Okay, sorry, 4 nitrogen is occupied by the nitrogen of the pyrrole ring, and the fifth valency is occupied by the imidazole nitrogen of the proximal histidine, and sixth valency is occupied by the imidazole nitrogen of the distal histidine. Okay, E7, histidine E7. But in the ferric state there is an absence of 6 valency. Okay, so there is a no question of binding of the oxygen. Usually whenever oxygen it binds to the 6 valency of the iron. So there is a no occurrence of the 6 valency. So there will be no transport, no attachment of the oxygen in that hemoglobin molecule. So there will be no transportation of the oxygen by that hemoglobin. Oxygenation doesn't occur. Okay, whenever the ferric is the oxidized state, but that hemoglobin is known as oxyhemoglobin. Okay, in this is very important. This line is very important. Okay, I am going to utter. In the oxyhemoglobin, oxygenation does not occur. Okay, whenever the iron in the heme in the ferric state, it is known as methemoglobin or oxyhemoglobin, isn't it? In the oxyhemoglobin, oxygenation does not occur. Okay, so that type of hemoglobin cannot transport the oxygen. Normally, the enzyme methemoglobin, anybody clear this part?
Yes, ma'am.
Why it is oxyhemoglobin? Because iron in the ferric state. In the oxyhemoglobin, this is the methemoglobin is the oxyhemoglobin. Okay, due to mutation of the gene. Okay, this oxyhemoglobin cannot be oxygenated, right? That is the thing. Normally, the enzyme methemoglobin reductase. Okay, methemoglobin reductase reduce the ferric iron of methemoglobin to ferrous iron. Okay, this methemoglobin can arise by oxidation of ferrous iron to ferric as a side effect of agent such as sulfa drug like sulfonamides. These are the sulfa drug, and from hereditary hemoglobin M, that is the due to mutation and consequence to reduce activity of the methemoglobin reductase. Okay, this methemoglobin reductase. This methemoglobin reductase, okay, required NADPH, okay, NADPH as a coenzyme for its activity, right? Methemoglobin reductase required NADPH as a coenzyme for its activity. HMP shunt or pentose phosphate pathway, okay, is the constant supplier of NADPH, major and constant supplier of NADPH coenzyme for hemoglobin reductase activity. That's all. Okay, that's why in HMP defect in the HMP shunt, okay, defect in the HMP shunt, that will be occurrence of the defect in the HMP shunt. There will be the possibility of the occurrence of the methemoglobinemia. Okay, methemoglobinemia. In hemoglobin M, what happened? What type of mutation? I am going to explain. In hemoglobin M, histidine, that is the proximal histidine F8, okay, F8 histidine in the F helix, 8th position histidine, always proximal histidine, it binds to the fifth valency of the iron, okay, that is replaced by tyrosine, okay, that is replaced by tyrosine. Tyrosine, it is a aromatic amino acid, okay, aromatic amino acid, okay, phenol ring containing amino acid, okay, tyrosine. The iron of methemoglobin forms a tight ionic complex with the phenolate anion, okay, with the phenolate anion of tyrosine that stabilize the ferric structure, okay, that stabilize the ferric form, okay. Normally that iron fifth valency is binds to the proximal histidine, okay, proximal histidine, okay, but in case of hemoglobin M, that is replaced by tyrosine, okay, tyrosine, but that tyrosine, the phenol ring of the tyrosine, actually tyrosine chemically para-hydroxyphenylalanine, okay, under the amino acid structure you are already told, I think tyrosine is a aromatic amino acid, that is the body can synthesize tyrosine, that's why tyrosine is non-essential amino acid. Body can synthesize tyrosine from phenylalanine. Tyrosine is the para-hydroxyphenylalanine, so phenol group is obviously there. The phenol group form the tight ionic complex with the, that is the in the fifth position, fifth valency of the iron, okay, that stabilize the ferric structure, okay, ferric structure Fe3 plus form, okay, ferric form. That's why the ferric to ferrous reduction, that reduction is not possible, okay, not possible. In alpha chain of hemoglobin M variant, RT equilibrium favors the T state. RT equilibrium favor the T state. Oxygen affinity is reduced and Vor's effect is absent. Okay, once oxygen affinity is reduced, obviously the Vor's effect will be reduced. Okay, until and unless oxygen affinity is not there, oxygen affinity, the force effect cannot be occur, isn't it? Beta chain of hemoglobin M variant exhibit RT switching and Vor's effect is therefore present, okay. RT switching is there, so oxygen affinity is intact in the beta chain, so Vor's effect is used to be there, okay. This one I am going to explain again. In the alpha chain, hemoglobin M variant, RT equilibrium favors the T state, okay. RT equilibrium favors the T state, okay. Oxygen affinity is reduced. Once oxygen affinity is reduced, the oxygenation of the hemoglobin is not possible, okay. Oxygenation in the lung, oxygenation of the hemoglobin, if it is not possible, okay, if it is not possible, there is a no chance of the release of the proton, okay. If there is a no release of the proton, then how the carbonic acid will form from the bicarbonate, okay. Normally carbonic anhydrase is ligate, okay, it responsible for reversible reaction, okay. Then proton attach with the bicarbonate to form carbonic acid, okay, normally carbonic acid, that effect cannot be possible, okay, it is cannot be possible, release of the proton cannot be possible until and unless oxygenation doesn't occur, okay. Mutation that favor the R state increase the oxygen affinity. This hemoglobin therefore fail to deliver adequate oxygen to the peripheral tissues. Okay, mutation favors the R state, increase the oxygen affinity. Okay, this hemoglobin therefore fail to deliver adequate oxygen to the peripheral tissue. Resulting tissue, this resulting tissue hypoxia leads to polycythemia. Okay, as a response of hypoxia, there will be excessive proliferation of the RBC. Erythrocytosis will be increased. Okay, that is the as a feedback response of the hypoxia. Okay, so there will be increased concentration of the erythrocyte. In the methemoglobin person, in the RBC count will used to be high. In the peripheral blood, RBC count will be high in case of methemoglobin. Now hemoglobin S, sickle cell hemoglobin. Okay, sickle cell hemoglobin. Hemoglobin S, the non-polar amino acid valine has replaced the polar surface residue of the 6th position glutamic acid of the beta subunit, beta polypeptide chain, generating a hydrophobic sticky patch. Hydrophobic sticky patch on the surface of the beta subunit. On the surface of the beta subunit of the both. Okay, remember this sticky patch. Whenever this glutamic acid replaced by the valine it is a non-polar amino acid, branch chain amino acid, isn't it? It is a non-polar branch chain amino acid. Okay, as soon as valine has replaced the glutamic acid, okay, there will be occurrence of the sticky patch. Okay, sticky patch on the surface of the beta subunit of the both oxy and deoxyhemoglobin S. Okay, both oxy and deoxyhemoglobin S. Both hemoglobin A and hemoglobin S contain a complementary sticky patch on their surfaces. Complementary sticky patch. Complementary sticky patch means where sticky patch is attached. There is a groove. Okay, there is a place where sticky patch is going to be attached. That is the complementary sticky patch. Where the sticky patch is going to be fit. Okay, fit means binding area for the sticky patch. Okay, complementary sticky patch means you can just imagine the binding area of the sticky patch. Okay, so hemoglobin A, hemoglobin S contain the complementary sticky patch on their surfaces that is exposed only in the deoxygenated state. Okay, this conformational change occur only the deoxygenated state, not oxygenated state. Deoxygenated when the hemoglobin is the T structure, deoxygenated. Okay, T form, tight form. Then this binding area means complementary sticky patch. Okay, of both normal and hemoglobin S is exposed whenever hemoglobin is deoxygenated state, right? Thus, the low partial pressure of oxygen, deoxyhemoglobin S can polymerize to form a long insoluble fiber. Okay, because deoxygenated state, that binding area is exposed. Okay, where sticky patch is attached, isn't it? Because sticky patch attach both oxy and deoxyhemoglobin, isn't it? So there will be formed. So bind with the attachment of the all hemoglobin, that hemoglobin is resemble to a resemble like a fiber. Okay, that fiber is insoluble because valine is insoluble. No, this is the branch chain non-polar amino acid. Okay, this twisted helical fiber distort the erythrocyte. This twisted helical fiber distort the erythrocyte into a characteristic sickle shape. Okay, so distortion of the RBC due to this fiber, appearance of the fiber structure. Okay, RBC become the fiber structure. Okay, it is attached to, because hemoglobin is attached to each other, isn't it, of the RBC. So characteristics of the, this look like a sickle shape. This is the hemoglobin 2 alpha and this is the oxyhemoglobin, this is the deoxyhemoglobin. Okay, deoxyhemoglobin and oxyhemoglobin. I have mentioned there sticky phase on the surface of the beta subunit of the both oxy and deoxyhemoglobin S. Okay, and both hemoglobin A and S contain complementary sticky patch on their surface binding area. Okay, this is the binding area complementary complementary binding area. Okay, complementary sticky paste, that is the binding area for the sticky paste. Sticky paste occur, as I have already mentioned, sticky paste occur on the surface of the beta subunit, both beta subunit, because this replacement, no, valine replaced by glutamic acid. Okay, replace the glutamic acid in the oxy and deoxyhemoglobin S. Once it is replaced, in the beta chain, it becomes S hemoglobin. Okay. Then deoxyhemoglobin also having sticky patch, and oxyhemoglobin also got the sticky patch. In the deoxygenated state also sticky patch, and oxygenated state also there is the, this is sticky patch is there. And complementary sticky patch, the binding site for this sticky patch present both in the hemoglobin S and hemoglobin A. Okay, both normal and S hemoglobin, isn't it? So whenever these all hemoglobins are all together, in case of deoxygenated state, okay, they are exposed, and their surface, both hemoglobin A and hemoglobin S contain the complementary sticky paste, that is the binding area for the sticky paste on their surfaces, that is exposed in the deoxygenated state. Okay, that is exposed to the deoxygenated state. Okay, because this one is oxygenated state, no, that therefore it is not exposed, because this is the deoxygenated state, this is also deoxygenated state, that's why it is exposed, isn't it? So in the deoxygenated form, as this complementary sticky patch area, binding area, that means exposed, so all these oxy and deoxyhemoglobin are get attached. Okay, there all are hemoglobin S and hemoglobin deoxyhemoglobin A and deoxyhemoglobin S are get attached, isn't it? This is attached here, and another deoxyhemoglobin A is attached here. They attaching each other, they will form the fiber like structure. Okay, once the fiber is formed, obviously it will fold to the helical structure. Okay, that distort the normal texture of the RBC to the sickle shaped. Okay, towards the sickling of the RBC. Okay, normal texture will be distorted, so there will be appearance of the sickling. Okay, sickle shaped RBC. Okay, this deoxyhemoglobin is combined to the deoxyhemoglobin S. Okay, this oxyhemoglobin also can combine here. Okay, in the S. Okay, like that. Then deoxy S also combine with here. Okay, like that it will be. There will be. But in case of oxy A in case of oxygenated state, okay, oxygenated state, it is not exposed. This binding area, complementary area is not exposed. So sickling is less. The possibility of the sickling will be less in oxygenated state. Okay, in the oxygenated state, in the normal hemoglobin also, or normal oxyhemoglobin A also, there will be no occurrence of the binding area, complementary sticky patch. And here also oxygenated state, there will be no exposing of the binding area, complementary area, that means of the sticky patch. So sickling, the chances of the sickling will be less, but deoxygenated state, there will be more chances of the sickling. Okay, because the expose of the binding area, that is the complementary sticky patch. Okay, you have to go through the Harper. Okay, to understand this concept. This is the concept. Here it is again mentioned, this complementary area, that is I have told the binding area, you can imagine as a receptor area for the sticky patch. Okay, where sticky patch is going to be attached or going to be bind. Okay, same thing. Biomedical implications. Okay, biomedical implication. Myoglobinuria. Okay, myoglobinuria. Myoglobin structure is similar like hemoglobin, but it has got only one polypeptide chain. Okay, only one polypeptide chain. Okay, so there will be no question of cooperative binding, as that cooperating binding doesn't occur. Okay, because of presence only one polypeptide chain, there is no question of Vor's effect. Okay, for myoglobin. Now myoglobinuria. Otherwise rest of the structure are same. Following the massive crush injury, myoglobin released from the damaged muscle fibers color the urine dark red. Myoglobin can be detected in plasma following a myocardial infarction, but assay of the serum enzymes provide a more sensitive index of myocardial injury. Okay, this one to all of you have known because clinical enzymology already finished. What are the enzymes for the myocardial injury? You have already mentioned. Okay, this myoglobin also can be detected in the plasma following the myocardial infarction. Okay, along with the rest of the enzyme. Okay, rest of the enzyme I am not going to repeat again because you have already finished the clinical enzymology class. Okay, anemia. Now anemia, what is anemia? Anemia is the reduction in the number of the red blood cell or of the hemoglobin in the blood, can reflect impaired synthesis of the hemoglobin. Okay, impaired synthesis of the hemoglobin due to the iron deficiency or impaired production of the erythrocyte. Okay, maybe due to impaired synthesis, erythrocyte production is normal but hemoglobin production is less due to iron deficiency, or as a whole in impaired production of the erythrocyte. Okay, whole erythrocyte is not, erythrocytosis is less. Okay, impaired production of the erythrocyte, that means erythrocytosis is the less in the bone marrow, isn't it, due to deficiency of the folic acid and vitamin B12. Okay, this result, if it is deficiency in the hemoglobin formation, that in that case also anemia occur. Okay, that anemia is hypochromic microcytic. Okay, any RBCs are very small. Okay, hypochromic hemoglobin also very less amount. It's because of iron deficiency. Okay, and microcytic. Microcytic means cells size of the cell is very less, very small. Okay, RBC hypochromic microcytic anemia. But as a whole, if it is impaired production of the erythrocyte, okay, due to lack of due to deficiency or decrease erythropoiesis due to deficiency of folic acid and vitamin B12, that is the macrocytic anemia. Macrocytic. Okay, cellular size, RBC size becomes larger. Okay, but in that case also anemia occur. Okay, defective synthesis of the RBC or defective synthesis of the hemoglobin. Okay, these are the two causes of the anemia. Okay, there will be reduction of the number of the cell or of the hemoglobin of the in the blood. Okay, that can reflect impaired synthesis of the hemoglobin or impaired production of the erythrocyte. Now thalassemia. Okay, thalassemia. See, don't confuse. These are the abnormal hemoglobin, isn't it? This sickles hemoglobin S, hemoglobin M. Okay, these are the abnormal hemoglobin. Okay, these are the abnormal. There are so many abnormal hemoglobin. I'll discuss in next class. Today it won't be sufficient time, enough time to go through. Okay, this hemoglobin M, don't confuse. This hemoglobin M and hemoglobin S, it is a qualitative hemoglobinopathy. Qualitative. Okay, not quantitative. Okay, quantity is okay only. Quantity is okay. Alpha chain and beta chain is quantity, as a quantity, as a number. Okay, number of production is okay, but defective production. Number is okay, but defect in the production, defect in inside. Okay, inside the chain. Okay, valine replace the glutamic acid in hemoglobin S, and there in the met hemoglobin iron is oxy iron, that is the ferric iron instead of ferrous iron. Otherwise production is sufficient. Production is sufficient, quantity is okay. Only qualitative defect. Quality is abnormal. Abnormal, that is a qualitative defect. Okay, abnormal hemoglobin, but number of the hemoglobin is okay. Okay, production is okay, number of the hemoglobin is okay, but quality is the abnormal hemoglobin. But here in the thalassemia, the quality is okay. Okay, hemoglobin is, quality is okay, but quantity is less. Okay, huh? Here in the hemoglobin M and hemoglobin S, quantity of the production of the alpha chain and beta chain is normal, okay, normal, quite normal. But there be synthesis of the abnormal beta chain, isn't it? Or abnormal heme molecule in case of hemoglobin M. Otherwise, quantity is okay. But here in the thalassemia, there is a quantity is less, amount is less. Otherwise, quality is okay. Normal beta chain and normal alpha chain, but quantity is less, amount is less. Okay, that is a quantitative hemoglobinopathy, thalassemia. Thalassemia is a quantitative hemoglobinopathy. Okay. The genetic defect known as thalassemia result from peripheral and total production is less, quantity is less. Otherwise, the hemoglobin as a structure, polypeptide chains are quite normal. Not like the qualitative hemoglobinopathy like hemoglobin S and hemoglobin M, right? There is a partial and total absence of alpha and beta chain of the hemoglobin. Over 750 different mutations have been identified, but only 3 are common. Either the alpha chain, alpha thalassemia, or beta chain, beta thalassemia can be affected. The superscript indicate whether the subunit is completely absent. If it is completely absent, alpha 0 and beta 0. Okay, no quantity, completely absent. And whether it is synthesis is reduced, a little bit reduced, then alpha plus and beta plus. Okay, quantity is a little bit reduced. Okay, that is alpha plus and beta plus. This one you have to remember. Now glycosylated hemoglobin, okay, HbA1c, okay, HbA1c, this is the different hemoglobin when glycosylated hemoglobin. When hemoglobin form glycosidic bond with the glucose, okay, glucose, that hemoglobin becomes glycosylated hemoglobin, When blood glucose enter the erythrocyte, it glycosidic bonds already mentioned, okay, glycosidic bond. What is glycosidic bond? Alpha or beta glycosidic bond, these are already taught. Glycosylate the epsilon amino group of the lysine residue and the amino terminal of the hemoglobin. The fraction of hemoglobin glycosylated. Normally about 5% is proportionate to blood glucose concentration, normal blood glucose concentration. Okay. Since the half-life of the erythrocyte is 60 days, total lifespan is 120, so half-life is 60 days, the level of glycosylated hemoglobin reflect the mean glucose concentration over the preceding 6 to 8 weeks. Okay, almost 60 days. Okay, 6 to 8 weeks. Measurement of the hemoglobin HbA1c therefore provide a valuable information for management of the diabetes mellitus. This is the chart showing the correlation between the mean glucose concentration, serum mean glucose concentration, serum or blood mean glucose concentration, and hemoglobin A1c in the patient with type 1 diabetes. Okay, this is the normal mean in non-diabetic individual, this one normal. This is the mean glucose in the patient treated with intensive insulin therapy. Okay, insulin therapy. Blood glucose concentration is 150. If it is 150, it corresponds to this one. This is the slope. This corresponds to the, say, 7. Okay, 6.9 or 7. Okay, if it is between the 200 to 250, this one is, say, 230, like that, it corresponds to the almost 9, like that. Okay, this is the mean glucose in the patient treated with started with insulin therapy. Okay, that's all about the various mutant variant of the hemoglobin. I'll also discuss some other mutant variant of the hemoglobin C, hemoglobin E, hemoglobin D, and Punjab phenotypes. Okay, these are also I'll discuss in the next class a little bit. Okay, any doubt?
No, ma'am.
This mutation of the hemoglobin, okay, this quantitative and qualitative deficiency of the hemoglobin, okay, this mutation of the thalassemia, okay, hemoglobin S, hemoglobin C, it acts Hemoglobin C not yet done, but it used to come as a short notes. Okay, and this glycosylated hemoglobin. Okay, thank you.
Thank you, ma'am.